Hello and welcome to brand new video of Pharmacokinetics. This is the part 1 of many upcoming videos on Pharmacokinetics series. I'm going to teach you each and concept in depth. So subscribe now. In this video, we are going to learn transport of drug across cell membrane, or you can call it biological membrane like a tissue or cell. So let's get started. What is pharmacokinetics? First, let's understand what is pharmacokinetics before we deep dive into various concepts of it. Let me explain you in simple language. When you take any medicine or drug, your body does some funny things to it which affect the drug and its therapeutic properties, such as absorption. Drug is ingested by the mouth and it is absorbed into body from gut, stomach and intestine. Once the drug gets absorbed, it is distributed into whole body via blood circulation. It is also metabolized by various biochemical reactions happening in your body. Then the last stage is excretion, which means the drug is completely excreted or removed from the body. Drug went through various events after taking it: absorption, metabolism, distribution, excretion. Pharmacokinetics is a study of absorption, distribution, metabolism and excretion of drug. That's what pharmacokinetics is. Transport of drug across various biological membrane. In absorption and distribution process, a drug has to move across various biological membranes like cell wall and blood-brain barrier, etc. The biological membrane is made up of two layers of phospholipid with intermingled protein molecule. All lipid-soluble substances get dissolved in cell membrane and they are easily permeated into cells. To understand mechanism behind it, you have to understand these three concepts: passive transfer, carrier-mediated transport, and endocytosis and exocytosis. Let's start with passive transfer. A passive transfer of drug is movement of drug across a membrane without any need of energy. Energy currency of cell is ATP, adenosine triphosphate. In passive transfer, cell doesn't have to spend any energy in form of ATP to transfer the drug. Drug is transferred in a direction of concentration gradient, which means from higher concentration to lower concentration. Outside of cell is a higher concentration and inside is lower concentration. Hence the drug is transferred from outside to inside. And once there is equilibrium in concentration on both sides, transfer is stopped automatically. Passive transfer of drug use basic laws of physics, which is movement from higher concentration to lower concentration to achieve state of equilibrium. In passive transfer, these are the two types of it: simple diffusion and filtration. In simple diffusion, drugs which are lipid soluble or unionized are transferred via simple diffusion. The speed of diffusion depends on the degree of concentration gradient, lipid solubility and ionization. Sometimes simple diffusion is also known as lipid diffusion because most drugs follow this as they are lipid soluble. In filtration, there are small aqueous pores on cell membrane which are also passage for drug to transport. Diameter of these pores is 7 Armstrong and aqueous soluble drug with molecular weight less than 100 can easily pass through it. Now note the difference between these two. Both simple diffusion and filtration transfer the drug across cell membrane in the direction of concentration gradient from higher to lower concentration. But simple diffusion is used for lipid soluble drugs only and filtration is used for water soluble drugs only. In simple diffusion, a lipid soluble drugs are easily dissolved or permeated into cell membrane because cell membrane and drug both have lipophilic properties. The cell membrane is made up of phospholipid, which is a lipid. On the other hand, in filtration, water-soluble drugs are transferred via aqueous pores because cell membrane are lipophilic as well as hydrophobic properties, which means any water-soluble drug cannot interact with cell membrane, and that's why there are aqueous pores which help in transfer of drugs. Next method of drug transport is carrier-mediated transport. There are some drugs or substances which cannot be transferred via passive transfer because every drug is different from its chemical properties. They need a special carrier protein which helps to transfer the drug. First, the drug is attached to the carrier protein, then the carrier protein takes it inside the cell and releases it, and the process goes on and on. That's how this mechanism works. There are two types of carrier mediated transport: active transport and facilitated diffusion. In active transport, the carrier protein has to actively use energy in form of ATP to transport drug across cell membrane. Active transport is useful when drug or chemical substance has to go against the concentration gradient, which means from low to high concentration to transport across cell membrane. Since this is going against the concentration gradient, there is expense of energy in cell and energy currency of cell is ATP. So cell needs to spend some amount of ATP to carry out this transport. In human body, natural metabolites such as sugar and amino acids, iron, etc. use active transport. Facilitate diffusion. In this, the carrier protein doesn't require any energy for transport, and the transport is in direction of concentration gradient, which means from high to low. So ATP is not required and it is energy independent. The carrier protein is highly specific, which means the binding site on carrier protein is specifically designed to bind with a specific drug or chemical substance at the binding site, and no other drug can bind with it if it's not meant to be. Example of this can be seen in glucose uptake of cells and absorption of vitamin B12 from intestine. If you look at the difference between active transport and facilitated diffusion, it is very simple. Both have same mechanism of work, but difference is active transport require energy and facilitated diffusion doesn't require energy. Active transport is for natural metabolites and facilitated diffusion is for very specific binding. For example, if a specific binding site is designed for vitamin B12, then other chemical substance cannot bind with it. But in active transport, any chemical substance can bind with it if it may belong to its natural metabolites. Now let's look at endocytosis and exocytosis, this method of drug transport. It is a simple concept. Endocytosis means the drug or chemical substance is engulfed by the cell membrane and carried it into the cell as a vesicle. When it reaches inside the cell, it is broken down and gets absorbed. That's how endocytosis works. Some anti-cancer drugs use this method for drug delivery to cancer cell. Exocytosis is reverse of endocytosis. Instead of engulfing in exocytosis, the chemical substance is expelled out or released from cell. Exocytosis process is very common in neurons when they release neurotransmitter which are stored in nerve ending. So that's it for today. I hope you learned something from this video, and if you need study notes regarding this video lecture, then the link is added in the description. Go check it out. Have a nice day. Bye bye.